Wait for a few more people.
Is Professor Go will join in?
I thought so.
Oh, sure. Then we will wait for him. Otherwise, I'll just go have a go.
Can we just wait a few more minutes, because there was over 25 that registered. So. Thank you, everyone that's already signed in. We'll just wait maybe another minute or so. All right, we might start, and if anyone else joins as we go, then welcome to them too. But welcome to everyone that's joined on tonight. I'm Jackie Henwood. I'm the Research Project Manager for CERA, so Centre for Eye Research Australia. I'd like to thank everyone for attending tonight's webinar, which will discuss tonight the retinal age as a novel, simple, non-invasive and cost-effective biomarker of ageing. This is workshop 9, so it's part of a series discussing artificial intelligence in healthcare, from data to algorithm to real-world solutions. These workshops are supported by the Medical Research Future Fund, and our goal is to connect the diverse skills of clinicians, researchers and healthcare workers with computer and artificial intelligence scientists. Therefore, we're trying to get everyone working together to develop and build AI solutions for real-world healthcare challenges. Before we start, I'd like to acknowledge the traditional custodians of the various lands on which we connect tonight via our computers, and pay our respects to their elders past and present. So tonight we're very lucky to have Dr. Lisa Zhu speak with us. So Dr. Lisa Zhu is a current research fellow within the Ophthalmic Epidemiology Department at the Centre for Eye Research Australia. Dr. Zhu's work has focused on the eye as a window of our eye health, which has created exceptional impact. So far, she has developed an excellent publication record with over 45 manuscripts, attracting more than 210 citations. She's given poster and oral presentations at international and national conferences, and her research program has attracted competitive grant supports, including prestigious NHMRC Investigator Grant. So without further ado, I'll pass you on to Lisa.
Thank you, Jackie, for your kind introduction. Then I'll share my screen. Oh, I'm not allowed to share screens. Sorry, Jackie.
Let me make sure that, can you now? Sorry.
Yeah.
Beautiful.
It works. Thank you. Okay. Can you see my screen?
Yeah.
Yeah, okay, thank you. Hi, everyone. This is Lisa. I'm a research fellow from Centre for Eye Research Australia. My research has focused on the potential utility of retinal imaging in predicting systemic disease. So I'm really honored to be here to give you a brief introduction on one of my projects, Retinal Age, a novel biomarker for aging. So in 2018, there was a TV show called Sisters Who Make Waves, went viral both in China and around the world. It is a singing competition show. There was a challenge out there in the social media to guess the ages of the sisters. And when you first see the video and you want to try to guess the sister's age, and the second video will show you the answers of each sister's age. And here comes the sister number 3. According to the perceptions of these two YouTubers, they guess the sister number 3's age is 32 or 35. Actually, the sister number 3 was already 52 years old, and they were really shocked, and they really wanted to ask her about the secrets of youth. So this is the warm-up for today's presentation, and the facial age, also known as a biological age. And the next term is the real age, also known as a chronological age. In this case, we know that the biological age can be quietly different from our chronological age. Biological age represents our functional decline, while the chronological age indicates the number of years that we have been alive. So currently we are in the aging society. According to the statistics, the number of people aged 60 years and older will more than double by the year of 2050. By that time, the number of the proportion of the people aged 60 years and older will account for nearly 22% of the whole population. Therefore, the healthy aging is a priority and the focus of the World Health Organization between 2015 and 2030. In the picture, in the right bottom shows that the systemic disease such as the heart disease, cancer, stroke, diabetes, and neurodegenerative disease increased significantly with age. So which means that the premature aging will undoubtedly lead to the great burden of the age-related disease. As I have shown in the warm-up, that chronological age can be quite differently from the biological age. So the aging process can be heterogeneous. For example, some 70 years old enjoying good functioning, while other 70 years old require significant help from caregivers on a daily activities such as dressing and eating. So the picture in the right side shows the 3 scenarios or relationship between the biological age and the chronological age. The horizontal axis indicates the chronological age in years, and the vertical axis represents the biological age. If our biological age equals to the chronological age, then we are in the healthy aging trajectory, as shown in at this line in this picture. And if our biological age is significantly larger than our chronological age, then this indicates our aging is accelerated, just like the pink area shown in this picture. On the contrary, if our biological age is significantly lower than the chronological age, which means that the age gap between the biological age and the chronological age is negative, our aging process is deaccelerated, which means that we are in a slow aging process. Therefore, the quantification of the aging process is very important, not only for the risk assessment, but also for the early individualized prevention and intervention. So far, we have proposed a lot of molecular, tissue, and cell aging biomarkers. The epigenetic age, which based on the DNA methylation status, is one of them. The epigenetic age gap is the difference between the epigenetic age and the chronological age, has been validated as a robust and reliable aging biomarker that can predict the future mortality. And also we have the image-based aging biomarkers such as the facial age and brain age. Previously, we usually derive the features such as the nose width from the facial image and the brain volume from the brain images to build the model to predict the age. However, recently, the introduction of the artificial intelligence, especially the deep learning algorithm, enables us with the advanced technology for the imaging processing, classification, and segmentation. So with the help of the deep learning algorithm, the accuracy of the brain age improved a lot. And what's more, the efficiency of the quantifying the brain age also improves due to without the necessary of pre-processing the brain images anymore. Also, the brain age gap, that is the brain predicted age, and the difference between the brain predicted age and the chronological age is also a predictor for future mortality. However, there are several limitations for the current aging biomarkers. For example, the high cost of the brain age and the invasiveness features of the epiclock and telomere, and also it usually takes at least 30 minutes to finish the brain scan. So these limitations prevented us from using these aging biomarkers for the population-based screening. And our retina at the back of our eye is a window to our whole body, given the similarities between our eye and the brain, heart, and kidney in terms of the genetic backgrounds, risk profile, and the pathology and the physiology. Therefore, studying and visualizing the retina can provide the insights on the health status of these important organs, such as the brain, heart, and kidney. In addition, the retinal imaging have the advantages of being cost-effective, non-invasive, fast, and safe. Currently, we are armed with a deep learning algorithm for better accuracy and efficiency in terms of the image processing. Taken together, the retinal age based on the deep learning algorithm might be a promising aging biomarker. So our study will be divided into two parts. The first part is to train and validate the deep learning algorithm for the age prediction. we have used more than 46,000 fundus images from the healthy population to train the deep learning model. Because there is no standardized way of quantifying the biological age, so we assume that the biological age equals to the chronological age in the relatively healthy population. And here we define the healthy population as those without chronic disease. And an independent 15 fundus images were used to validate the deep learning algorithm. The deep learning algorithm performance was really good on the age prediction. The left picture, the picture in the left side indicates the strong association between the predicted age and the chronological age with a coefficient of 0.83. The closer the coefficient to 1, the better performance. And the picture in the right side indicates the averaged deviation between the predicted age and the chronological age is 3.55 years. The less the MAE is, the better the performance. We also explore the contribution of each pixel to the age prediction, and we found the regions around the retinal vessels have been highlighted. So in terms of the accuracy of the retinal age, our model achieved the MAE of 3.55 years, which outperformed almost all the existing aging biomarkers. For example, the epi clock with an MAE of 4 to 5, blood biomarkers with an MAE of 5 to 6, the transcriptome's age with an MAE of 6 to 8, and also the image-based biomarkers with an MAE of 4 to 7 for the brain age and 3 to 6 for the facial age. In addition to the accuracy, the retinal age have the advantages of being fast, safe, cost-effective, and user-friendly. These advantages enables the retinal age is a much more accessible screening tool for aging risk assessment. So in the second part, we wanted to validate the clinical implications of the retinal age gap. That is the predicted age, that is the difference between the predicted age and the chronological age. So a total of 8,212 participants from a cohort study were involved in these steps. And the trained deep learning model processed all the retinal fundus images from the right eyes of these participants and predicted the retinal age. Then we calculated the retinal age gap, that is the gap between the retinal predicted age and the chronological age. When the retinal age gap is positive, which means that the retina appears older than the chronological age. And on the contrary, a negative retinal age gap indicates a younger appearance retina. So currently we have followed these participants with more than 11 years. So the data deaths can be recorded, and then we can link the retina age gap and the future risk of mortality. This slide shows the relationship between the retina age gap and all-cause mortality. The horizontal axis represents the retina age gap, and the vertical axis represents the risk of the all-cause mortality. This picture shows that the relationship between the retina age gap and all-cause mortality showed a J shape, which means the all-cause mortality risk increased significantly over the retina age gap. So in summary, the retina at the back of our eye is a unique window to our body and aging. The retina age can be accurately predicted by the deep learning algorithm with high accuracy, which is outperformed with the existing aging biomarkers. And the retina age gap, that is the difference between the predicted age and the chronological age, is a robust aging biomarker that can predict the future risk of mortality. And in terms of the future utility of the retinal imaging, I think it could be used as a screening tool for aging risk assessment. And also, given the similarities between the eye and the systemic disease, maybe the retinal age could be used as a biomarker for the future risk of important life event, for example, the cardiovascular disease and the neurodegenerative disease. And I would like to thank you to all the team members in our research group, and not only from the clinical group, such as the Professor Mingguang He and Dr. Dan Li Shi, Dr. Wei Wang, and Dr. Zachary Tang, and Dr. Wenyi Hu, and also the technical group, Professor Zongyuan Ge, Mr. Guang Kaipeng, and Mr. Wei Meng. And that's all. Thank you for listening.
Thank you, Lisa. That was very interesting. Now, did anyone have any questions? We'll just see first. And all right. Well, I think you actually covered a lot of it there. And I was going to ask you, once with the retina age, I was going to ask you what happens in the future, but you talked about there at the end, you touched on future plans there, and will you be integrating this with some of your future studies?
Yes. Currently, our study was based on the UK Biobank, which is mainly a Caucasian population. So the generalizability of this algorithm may not that well in other populations, for example, the Asian population and the African American population. So we wanted to improve the generalizability of this algorithm with more data inputs, for example, including the fundus images from the African American people and the Asian people. And that would be very good if we could get those images and improve the generalizability of our model.
And was there any other questions? Oh yeah, you just stopped sharing screen there. I was like, oh, you jumped.
Yeah.
Yeah, well, I think it was quite a comprehensive talk there, Lisa. And you have spoken about the future projection, which I think is very interesting as well. And I think that's, does anyone else have any other questions, or have we covered them for tonight? All right, well, I think we might leave it there.
Yeah, okay.
Yeah. Well, thank you for your talk tonight, Lisa, and thank you for everyone that, oh it was just a great talk. Thought it might be a question. I thought someone just got in there with a question.
Thank you, Catherine.
Beautiful. And I'll give everyone just a minute or so in case I think of anything. All right. Yeah, we'll leave it there. Well, thank you, Lisa. Thank you to everyone for joining in tonight.
Thanks for having me.
Yeah, and it was great to have you. And the next one will be the 19th of April. So definitely if anyone can tune in for that one as well, there'll be links and advertising sent out. So thanks everyone for joining, and that's everything for tonight.
Have a great night. Bye.
You too. Thank you. Bye.